Hello and welcome yet again to Effortless Mastery. Today we'll be learning on cell division, but we'll be focused on meiosis. Now remember in our last class, when we had to talk about cell division, we said it just how does the body move from a zygote, which was just one cell at the start of life, to a full-grown organism that has as much as 10 raised to power 13 cells. How did we move from one to so many? A process called cell division. And we established in our mitosis video that there are two types of cell division, being mitosis and meiosis. So please, if you are yet to see our video on mitosis, go back and check it. It is on this particular series. Now, today we'll be learning meiosis. What is meiosis? Meiosis is a type of cell division whereby the parent cell divides two times. It's two times. It will divide first and ultimately divide into four daughter cells. So meiosis is a type of cell division where the parent cell ultimately divides into four daughter cells. These four daughter cells are unique. They are genetically unique. That means they are not identical. So they don't look like each other. The four daughter cells differ in different things. We call it genetic variability. So there are four daughter cells that have some variabilities. They are not similar entirely. Now, and a key thing there is also that there is a reduction of number of chromosomes from diploid, which is 2n, to haploid number of chromosomes. So I repeat myself. Meiosis in simple terms is that it's a type of cell division whereby the parent cell divides into four daughter cells which are unique. That is, they are genetically variable. They are not the same thing. And these people have something similar. There's a reduction in their chromosome number from the regular diploid number we see in a normal cell to an haploid number seen in these cells. Meiosis occurs in germ cells. These are cells that will form sex gametes. Cells that will form sex gametes used for what? Procreation. So the essence of meiosis is really for the future. Unlike mitosis, which we said is for growth and tissue repair, meiosis is really about procreation that is done at fertilization. So when I decrease the number of chromosomes to haploid for the sperm and haploid for the egg, when they fuse, they form back the diploid, which is the zygote. All right, now the stages of meiosis is simple. Now, first and foremost, meiosis usually is divided into two. So I'll have an interphase. After the interphase, which is regular, I'll now be having first meiotic division and then second meiotic division. So during the interphase, the regular things we knew happen. Please, you can check back on mitosis video to understand better what happens in interphase. But we summarize that there are three phases or substages under interphase. The three of them, number one is called G1, where there is growth. So the cell I have just grows in size. In G2, in A's phase, the genetic material is duplicated, DNA is duplicated. And then in G2, which is still all under interphase, you realize there's maturation, there's double checking. Any mistake in the DNA is corrected. All right, so after interphase, I come straight into first meiotic division. And the first meiotic division is similar, like it's going to have the same steps we know about, which is PMAT. But the only thing here is that the P, which is prophase 1, is actually a very long step. In fact, it's the longest step or longest stage of meiosis. So it is subdivided. So this makes it different from the regular person we know. It's subdivided into 5 steps, which is leptotene, zygotene, pachytene, diplotene, and dikinesis. So what happens in each of these steps? Because it is important we know. Now, in prophase 1, which I said is the longest, first and foremost, remember in interphase, how does your cell appear? There's usually a centromere, single centromere. All this was mentioned in our mitosis video. Centromere. And then you see that your chromosomes are actually thin. They are not visible. And then there's a nuclear membrane. So what will happen in prophase here? The first thing in leptotene is that condensation of chromosomes begin. So the chromosomes begin to condense. They become visible. So of course, when they are visible, what are you going to see in a chromosome? Because of the duplication that occurred in interphase, you'll be seeing two sister chromatids joined or held together by a centromere. Centromere. All right. So that is what we will see. So of course, you use this picture, you see one. So this is a chromosome, this is a chromosome, this is a chromosome, this is a chromosome. Of course, there should be how many in a normal cell? 46. That's 23 pairs. So these are all of them here. So I could draw 4 on the board. Now, what happens after phase 1, which is condensation, making them visible? In phase 2, called zygote. In zygote, 3 things happen. Or would I say a particular stuff happen, which is a process called synapsis occurs. What is synapsis? Synapsis is just the pairing up of homologous chromosomes. So the word homologous, it's a Latin word. Homo means same or similar. So I just pair up, out of the 46, I pair up similar chromosomes. So that means from 46, I'll be having 2 sets of 23. So if we'll be having 23 pairs, 23 pairs. So I just pair them up. That pairing up is a process called synapsis. And when I pair things, remember each of the chromosomes should be having 2 sisters. When I pair them up, I will now be having chromosomes. There's 2 chromosomes joined together. And how many sisters? 4 sisters. So for that reason, their names are now called tetrads. So you realize in zygote, I like using STP to remember there is synapsis, a process called synapsis. What's synapsis? Pairing up of homologous chromosomes. And once they are paired up, what do I form? Tetrads. All right, the next step under prophase 1 is called packaging. What happens in packaging? I use the word CC for it. The first thing that happens in packaging is that there's a process called crossing over. That means because these guys that were joined together, these 2 homologous chromosomes were joined together, at some point, there will be transfer of genetic material. Those points of transfer, that is where I have maybe chromosome A sending something to chromosome B, exchanging genetic material to chromosome B, is called chiasmata. And that process of exchange is called crossing over. And this is one of the things that is responsible for the genetic variability. Why they are not similar? Because there is crossing over. Now, beyond this, another thing that happens is in diplotin. Diplotin, key for the focus of what we are learning. The only thing that happens in diplotin that is important is that the chiasmata will gradually be shifting towards the end so that it could be ready to divide. It will be shifting towards the end, the terminal point of your chromosomes. Another key thing about diplotin, which we will learn when we talk about oogenesis, which is development of the female sex gametes, is that it is the stage where the primary oocytes are arrested at birth. It is that stage they are arrested. Then dikinesis here, of course, the other things we know usually happens in prophase will happen here. There's disintegration, formation of mitotic spindles will start happening. All those things we know should happen will happen in dikinesis. And that's your chiasmata keeps shifting towards the end. So after prophase, we should have metaphase. What's metaphase? This phase will be aligning in the midline. And then anaphase. Anaphase will now be what? A split. But it is important to know the split in meiosis. In mitosis, the split was at the centromere. Remember, they were connected to the mitotic spindle was connected to kinetochore. So when I split, I divide into 2 sisters. But right here, when you split in first meiotic, that is anaphase 1, you are actually splitting the paired chromosomes into 2 individuals. So that means they were 46. I paired them up into 23. When I split them, I'll split 1 chromosome go here, 1 go here, 1 go here, 1 go here. That now leaves me with 23 to one person and 23 to the other person. So that's how we have haploid number after phase meiotic. You remember what happens in telophase according to our previous video on mitosis. So it's just the opposite. From condensation, you now be having diffusion. It's the opposite of prophase. So all the things that happen here will just be reversed. The nuclear membrane will be reformed. No duplication. Mitotic spindle will now do what? Disappear. So that's what happens in telophase. Now the next thing is now meiosis 2. Now immediately after meiosis 1, forming my haploid numbers, the cells go directly into meiosis 2 without doing another interphase. So what happens in meiosis 2? The meiosis 2 is the exact thing, or what I say is very similar to mitosis. So if you remember in mitosis, what happened in mitosis? We had prophase, metaphase, anaphase, and telophase. The prophase here in meiosis 2, which is the same thing as mitosis, would have 3 processes or would I say four things: C, D, D, F. The first alphabet C is that the chromosomes will become will condense, so they become visible with two sister chromatids. The D here, first D, is that the centrosome will duplicate and migrate to opposite poles. The other D is that there is disintegration of the nuclear membrane, and the F there is that there is formation of mitotic spindle. So metaphase alignment yet again, alignment. And please remember that when the mitotic spindles are coming here, they are attaching to kinetochore, which is a part of the centromere of just one chromosome. In meiosis 1, prophase 1, when they are coming, they are attaching to one chromosome here, one chromosome. So the two chromosomes that came together to be a pair, I will attach on one end, attach on one end. So that is that for that. For the metaphase, anaphase, they split. The split here is at way the centromere. So to split at the centromere, I'll be having two sister chromatids. Telophase will be the exact same thing. So if you want to fully understand this, of course, just refer back to our mitosis video. It's the exact same thing. So in summary, meiotic division or meiosis is a type of cell division whereby one parent cell, which was a diploid cell, divides to form four daughter cells. These four daughter cells are unique and they are genetically variable. So they are not the same exact. That's why even when we are dealing with the same parents, you can have the same exact kind of children. Now, from that, we also have that the number of chromosomes is reduced from diploid number to what? Haploid. So you can see this was diploid. This is now how many? From 2n, I'm now having n. That's haploid number. Now, it occurs in germ cells, and then it is for the formation of sex gametes, which is essential for what? Procreation. Now, of course, it's similar to this thing. There will be an interphase, but there will be a first meiotic division and a second meiotic division. So the first one will just be prophase 1, metaphase 1, anaphase 1, telophase 1. This one will be prophase 2, metaphase 2, anaphase 2, telophase 2. Now, what happens in meiosis 1? The most important step, which is the exact difference between meiosis and mitosis, is prophase 1, where here we have 5 substeps: leptotin, zygotin, pachytin, diplotin, and dikinases. In leptotin, there's condensation. In zygotin, there's synapses, which is pairing up to form tetrads. In pachytin, there's crossing over. Diplotin is really key in oogenesis because it is where primary sites are arrested. And that's that. And we have dikinases. Then other steps are the exact same thing. Metaphase, these guys will align. The mitotic spindle will be attaching to the kinetochores. But this time around, since they are paired up, I'll attach to one, I'll attach to one. So when I want to split, so the way sister chromatids, when I want to split, I split this way. So one chromosome goes here, one chromosome with its two sister chromatids. Remember in mitosis, it is one person dividing into two. So that means sister chromatid, sister chromatid. All right, so that is that. It just happens to the end. And once we have haploid people, meiosis 2 in short, which is similar to mitosis, we'll do the exact same thing and we get here. Thank you very much for joining us. I believe that you fully understood meiosis. You fully understood meiosis. To stay connected with premium, to receive premium lectures, stay connected to this channel. Ensure you subscribe, like the video, and then comment in our comment section so that this video can go to as many people as possible. Ensure you also share the video. Thank you very much for joining us. I will see you next time when we continue in this series on general embryology.